Welcome to another episode of the Synthetic Biology series. We are NTU-Singapore, and today we will be going through diagnostics and why it is so important in today's world. So, why is diagnostics so important? They help us find out if a person has been infected with a virus or a disease. Diagnostics can help to provide accurate and specific information that is essential in detecting diseases. Some of the conventional diagnostics available are usually tests done in labs, such as using a microscope or conducting immunological tests. Some advantages of these tests include detecting a particular stage of the virus life cycle and identifying its phenotypic characteristics. However, these methods are time-consuming, and for different stages in the life cycle of the virus, its characteristics might differ, which leads to multiple tests required. Generally, conventional diagnostics starts with microscopic identification, followed by microbiological identification, where the researchers will try to identify the behavior of the virus. From there, we will be able to differentiate the virus types and lastly, conduct immunological assays to identify antibodies that can bind to these viruses. There are concerns about just relying on conventional methods. Over the past few years, new pathogens have emerged, such as the COVID-19 virus and Ebola. In addition, new COVID-19 variants, such as the Delta, Delta Plus, and Mu variants have recently emerged. Lastly, bioterrorism, where terrorists try to spread new viruses, is a big concern in the future. Therefore, it is essential to have rapid diagnostics to prevent the spread of new viruses. This is where SynBio-enabled diagnostics comes in, also known as nucleic acid-based diagnostics. These are used as a point-of-care service where clinicians do not need to wait long hours before obtaining the results. This is made possible by using specific primers or probes to detect the virus. In general, developing these rapid diagnostics starts from identifying the target genome. This is done by sequencing the whole genome. Next, primers or probes are designed to identify the sequence for amplification. Once it is amplified using commonly used techniques such as the polymerase chain reaction, PCR for short, it can then be quantified using detection methods such as fluorescence. The main difference between conventional and SynBio-enabled diagnostics is that conventional diagnostics require multiple tests to test for different variants. In contrast, SynBio-enabled diagnostics require only one test to detect numerous variants. Some examples of synthetic biology-enabled diagnostics include the COVID-19 multiplex assay, which was the first COVID-19 diagnostic kit, and the diagnostics to detect MRSA. Lastly, we have the DNA origami nanostructures that can be used to detect malaria or heart failure. However, this is still under the research phase. So now that we have learned the progress of diagnostics in the field of synthetic biology, how important do you think diagnostics will be to you in the future? That's all for today, and see you soon in the next episode.